Dr. Leone, we see your slides. We have not heard you yet. I think you're still muted.
I have to unmute him. Okay. Hi, everyone. Thank you for the invitation. It has been a long time, since a couple of years since I attended this meeting. And I'm very excited about it because here we are 26 years after I began the first gene therapy trial for Canavan disease. And in the evolution of this project, I met incredible investigators, wonderful people, families, foundations, and that definitely changed the direction of my career in so many ways. But today I'm here to just recap what has happened during the past 26 years. And also, this is designed for families most than anything else. I thank the NTSAD for inviting me. I thank all of the speakers for giving the great view and update on Canavan disease. But what we have here, let's see if I can remove this from, what we have here is pretty much the evolution of this. And what I would like to bring attention is that 20 years ago, just about this month, in 2001, we were given approval by the FDA for the first time to use AAV in the brain for a neurologic disorder. And this happened exactly 20 years ago. So 20 years now, I'm just starting a new clinical trial after 20 years of knowledge, 20 years of natural history data in patients, 20 years of animal models and cocktail of medications and everything that's happened between, and many publications and a lot of hard work from my point of view and many others, of course. So it's very exciting. We have started. This project actually took a foundation 3 years ago when we moved into manufacturing and preclinical studies with the primary objective to test safety and efficacy in patients, in Canavan patients. So, so here we have pretty much a recap of the AV2 studies. Many of you have seen it. Some have even participated, some of the families that I know from the NTSAD community. So this, what we've done in 2001 would never have been possible without the creative mind of Dr. Samolsky and Zhao Zhao. So they developed a way to actually purify vector without wild type viruses. And so that was the way. And so that's, we moved from liposomal transfection, which is not that efficient, to AV2. That was the greatest. So I would like to show this. There is part of everything that I'm citing, that I'm reporting here has been published. What we use as a design, experimental design of the AV2 study is similar to what we are using now. For instance, the pretreatment phase with the MRIs and an MRS to measure NAA. As you all know, in Canavan, there is an elevation in NAA. You have heard it multiple times, and why? Because of the mutations in the inactive mutations in the Canavan gene, and neurologic examination with gross motor function measure, Ashworth scale for spasticity, Mullen, PDI, pediatric disability index, and other tests that we do. So this is all being published, and as you can see, and you can find it online or here, this is very straightforward with the pretreatment phase, a surgical phase, and a post-gene delivery phase that lasts really a lifetime. For design, 2 years, but really any Canavan patient that really undertake gene therapy, any gene therapy patient just is going to be followed for the lifetime. And that's the responsibility that any sponsor and investigator takes when just submits an IND that is clear for clinical use. And natural history, we have 26 years, and a lot is published, some is not. But from what we can see here, as I said, everything I'm presenting is published, is that in the natural history of Canavan disease, we have millimolar increase in NAA in the brain in several areas. these data are published, and this is like a typical peak. Any neuroradiologist will look at this peak and will say, here is Canavan. And then from then, everything else, all the studies that every family here probably has gone through, and I feel for you. So the brain mass here, as we all see, once again, up to 5 years is reduced. We have tons of data, a lot published. I'm not gonna just spend much time on this because you can find it online, and this is it, is 20 years of data. What we have here, we're looking also at T1 relaxation time during the first 2 years of life, during the myelination period. T1 relaxation should be reduced. Here is clear there is abnormal in Canavan. We use it as an index of myelination, even though should be more of lipid and water content. In any case, a lot of abnormalities in the brain of Canavan patients. And we also demonstrated with pre-treatment follow-up of our patient that edema precedes progressive atrophy. And this was demonstrated in 1967, by the way, by an investigator that was funded by the NTSAD. So that's pretty impressive. So what did we do with the first gene therapy? We targeted the neurons because that was a type of serotype, AV2, the most common that we could use then. We were the first, after all, and that was the safest. So we also targeted neurons because the compartment that, as Dr. Pleasure mentioned as well, that synthesize NAA is in fact neurons. So we wanted to reduce NAA neurons, and we did significantly in the frontal and periventricular area, where we also saw some myelination, also the yellow, just a shade here is just would be normal NAA in age match control. We did reduce brain atrophy post gene therapy. We stabilized hydrocephalus, and we also saw some improvement in gross motor function measure. In some of the patients, not all of the patients, was statistically significant at 18 months post gene transfer. We saw a reduction, actually resolution of seizures, and that really replicate animal studies, many animal studies that have demonstrated this. With gross motor function measure throughout these 20 years, we can definitely have a model of prediction of Canavan disease, also looking at the mutations and combination of mutations and polymorphism that sometimes appear. So a typical Canavan patient, when is tested with a gross motor function measure, a standard test that a neurologist or physical therapist can do, it just has 5 dimensions. And typical Canavan patients do not just score anything typically beyond lying and rolling. So that's what we have, 98%, we have it. And that's for every other test that just the test specifically motor function. For cognitive tests, in fact, Canavan patients are much more developed than motor function. But when we look at motor function and myelin, involuntary movement, that's what we're analyzing. With a typical patient, and we have had, and many are published as well, and I've met many of them, and we tested many of them, is that they can reach the sitting and crawling and kneeling. And then there are the atypical Canavan with minimal or no symptoms, which we published on in 2006 in Annals of Neurology, in which 2 girls, 2 sisters are pretty much developing normally the driver license, they're going to college, and they were just diagnosed with Dr. Kolodny, we refer the patients to him because one of them had seizure, but the pediatrician was very smart, saw that something maybe was off, and that was it. We found a new mutation, which was actually tested, also found in other patients that have an atypical form, a completely atypical form of Canavan disease. This is a predicted trajectory of disease progression 2021, 2022. It's April 2021. There are 6 new Canavan patients that are typical because of we haven't had the GMFM score, but I've seen them. So just it's easy to score a child that is 6 months old or a year old that cannot hold the head up and has a GMFM of less than 5%. It's very easy. But in any case, with that said, the prediction is that within 2021 and 2022, there will be 12, let's say, that will score like this, so with less than 5% function. And we have one, for instance, that is atypical and will score here. So that's just a predictive trajectory. It's not data. I have the data, but I am not presenting them today. So what's important here is also collaborate with investigators and in 2017, this great work, the genotype and phenotype correlation in Canavan disease in patients that in fact are atypical, and they are atypical because they have tiny residual activity. This patient does not show the 1% more activity because only one mutation was tested. This is just very technical. It's obvious that the patient is normal. I mean, normal language, walking, and sitting and running. So without going into much of a technicality of it, was clear to us that targeting NAA, reducing NAA at the source by expressing aspartate in neurons which normally do not express was not the right strategy. So we started looking into targeting oligodendrocytes, but we could only do it when technology advanced. And that was done with the great collaboration of Steve Gray and Tom McCown at UNC and Drew Samosky. We developed this oligotropic vector, the first oligotropic, natural oligotropic. We didn't need to restrict any promoter. And so we started working on it. First demonstrated that, in fact, we had an effect, not only in myelination, but also on all the other markers that we published in the past 20 years on obviously just energetic demand in myelination. There is like an astrocytic vacuolation that is present in Canavan disease brain. So this started a path. There was 2016 that we published it, and then in 2018, great work from Dr. Klagman and collaborators about mice engineered to make elevated NAA that did not show Canavan-like pathology. So the high NAA in this model in wild type does not cause Canavan. That's because when Dr. Pleasure mentioned, and there is also Morris Baslow that you may have remembered, just like the idea that the 3 compartments, in fact, are working like neurons, astrocytes, and oligodendrocytes in this disease, and that really the pathology is caused by high NAA in the context of no ASPA. So what the team did is that they engineered to lack ASPA only in oligodendrocyte, and they do show, in fact, they did show Canavan-like pathology. But the authors hypothesized that oligodendrocytes without ASPA are uniquely sensitive to elevated NAA. This confirmed the actual hypothesis, Dr. Pleasure hypothesis, that 2 wrong make it right. And so we moved on, and there was the 2018. This is when we started this project, and we're now in the clinic. We just had the surgery in the first kid 15 days ago, and just brilliant. We're thrilled to just give you the wonderful news. So we just published this amazing just a work on biodistribution, route of administration. We compare AV9 with AV olig because we wanted to see in the same model head to head how the 2 would work. And this is all published, is visible. Just we rescue vacuolation with the oligotropic at all of the dose, at the higher dose, not with the AV9. We rescue the phenotype in terms of motor function. The blue line is in fact the oligo-ASPA at the higher dose, which is significantly different than the sham, which means the Canavan mouse that had no gene therapy. So with this said, what I have today is just I would like to show you what we have going on, which in a second should go on. For some reason, it's not clicking. Okay, let me see. So I'm not sure if there's something. Okay, I got it. Okay, so what we have was disappearing because I have a big doc here with all your faces. Hi, everyone. So we have our CAN GT. we can scroll down. The sponsor is a company that just formed in 2019 so that we could speed up manufacturing and just submission of the IND for so many reasons. And here we are. What we have is the condition is Canavan disease. We have the drug, which is called AAV olig ASPA, generic name, also has a brand name. So we specify why we use the ASPA, because we target oligodendrocytes. So we're going to enroll 24 children, 24 participants. The trial is open to everyone. There's not going to be a control group. We can never have a control group in this type of gene therapy trial because of risk-benefit ratio. And the other thing about this application that is very important is that, number one, the AAV olig is not really great in transducing the brain if it's injected IV. And the other issue is that if we injected IV, we would have a limitation on the size of the patient, like there have been many other examples out there, for instance, in the spinal muscular atrophy, where the maximum age is 24 months because after that, body size increase, and so the vector would be diluted and would not be feasible and manageable, I guess, from a business perspective. So what we have here that we're injecting is 370 trillion viral particle of a pure GMP product that we made overseas because it was the best lab manufacturing facility that we could find. We had R&D, research and development that went very well. So with that, of course, we just prescribe just antiepileptic for a period of time just to avoid any issues. And this is typically normal after brain surgery. Very low steroids, just because we better just avoid any immunogenic response, even though this capsid is completely novel and would not be recognized by any antibody that is present. But just to be safe, and in fact, we may actually remove the steroids after a couple of patients. And in terms of just safety evaluation, similar to what we have done.
Dr. Leone, I'm just going to interrupt you because we're pretty much out of time. So if you want to have another minute or two, just to your very last minute or two, and then we do have a question at least, and see if there's any other questions who come in. So I wanted to save time for some questions.
Okay, I just want to acknowledge all of the people here, just to say that we're working at Dayton Children's Hospital, and this is the team of program manager and patient advocacy, Drew Samolsky, that without him, none of us would be here. And also wanted to thank our first patient who just left the hospital after 48 hours, 3 days after surgery, just 2 weeks ago. And here are the two just dog tours of the PICU in Dayton Children. Thank you.
Great, thank you. And you spoke about your vector AAV, the oligo one, being novel. Can you sort of compare this for our families to the research with AAV9 and give us some information about how they're different?
So it's all published, but in terms of differences, there are other labs that have done quite a lot of work. AAV9 is not specifically efficient in transducing oligodendrocytes. We know that through IV can just be transduce genes in ganglia, in primary neurons, in neurons, in motor neurons, in astrocytes, but not particularly efficient in oligodendrocytes. And we just is published, and it's just information that we can find anywhere. But we had to have the head-to-head comparison, and for that we need to have the same animal model, publicly available, just obviously very well characterized, and many people, in fact, Dr. Preja also work on that on the same mouse developed by Traca. And so head-to-head, same dose. We look at the transduction, and we saw, in fact, that was not efficient AAV9 expressing ASPA on transducing in oligodendrocytes, and that reflected in rescuing histopathology, which is typical in Canavan disease with vacuolation of the brain. And and for that, obviously, just our paper demonstrated that the AV olig is the way to go, at least at the moment. I mean, I would be even open if we look into a way of bypassing immunogenicity, even having the two therapies together. But that is in the future. But right now we have 24 patients with a second batch of vector that is being produced, and I'm like absolutely thrilled to tell everyone that this is the news, and it's a wonderful news for everyone at NTSAD and our Canavan community.